So we're continuing the chapter. This is all talking about things getting into and out of living things, mostly the cells, through the cell membrane. The cell membrane, you remember, is lipids. The lipids are kind of gapped out, so some things can get through the gaps between the lipids, but for the most part, the lipids are a pretty waterproof barrier, and the only way for stuff to get through the membrane is for the membrane to somehow let it through. Sometimes by putting pores in the membrane, we talked about those, and sometimes by grabbing stuff and moving it. That's what we're talking about next. If stuff is getting through the cell membrane, because that's the way it's going to go, is that the plants are using water for photosynthesis, and that's moving water up and into those cells, then that is something that the cell isn't really doing, it's just happening. That is passive transport. So transport is going through the membrane, but the cell isn't really exerting any energy to move it. Now, sometimes you will see active transport, the opposite of this, defined as the cells use energy to do this. But that's not a great definition for it, because the cells sometimes need to help things that would go through the membrane, but the membrane isn't really permeable to them. So the cell has a carrier that will carry stuff through the membrane. And it'll go, in a lot of cases, it'll go either way. So if there's more on the outside, it's going in through the carrier. If there's more on the inside, it's going out through the carrier. But there are carriers that only work in one direction. They'll let stuff in. So if the stuff is out there, they'll let it in, but they won't necessarily let it back out. And so there is energy involved in some of these things. So it's not as passive energy-wise, but the cell isn't really making stuff go. That's active transport. We talked about pumps. If the stuff is going a different way than it should, just like we pump water against, the water wants to sit there, we want to get it up, is that it takes energy to do that. You need a motor and a pump and electricity or whatever you're running the pump on to do it. The same thing's happening here. There's way more of this stuff on the inside than is on the outside, but you need it, so you pump it in. And that is active transport. And you can recognize it because it's going from an area of low concentration to an area of high concentration. It's going from low to high. It's going uphill, like you would have to pump stuff uphill. There's a type of active transport that's a little trickier, where there's stuff on the outside that you need inside, or stuff on the inside that you need outside, but it won't go through the membrane, and you don't have a pump, and you don't have a carrier for it. So what you do is you use membrane-enclosed bubbles to, in this case, you take stuff that you want to have out and you put a bubble around it. You move the bubble up to the surface. Now remember that the lipids kind of flow together like two soap bubbles meeting and becoming a bigger soap bubble. And so once this bubble hits the surface, it opens up as it flows into the surface, and whatever was in there is now out. This is called exocytosis. I'll write it down in a second. And the opposite is true too. Sometimes there's stuff on the outside. Maybe it's a prey animal or a piece of bacterium that you want to have in, that you're going to digest, or it's just material that's out there that won't go through the membrane, but you need it. And your membrane detects that it's there, and it will actually fold the membrane in. There's proteins here that'll fold the membrane in. And these things will go into that bump, and the bump gets pinched off, and now it's a separate bubble inside the cell. And maybe it can be broken down there, maybe the bubble can be disassembled, and then it releases it inside the cell, but this all takes energy. So this goes under the heading of active transport. Endocytosis stuff going in, and exocytosis stuff going out. They used to call endocytosis phagocytosis, P-H-A-G, from a Latin word for eating. And they would see it happen, and there was nothing, they couldn't see anything inside the bubbles, so they went, oh, the cell must be drinking. They called that pinocytosis. But then eventually people realized, no, there's stuff dissolved in that water that it wants in. It's not pulling the water in. It's pulling stuff that we can't see through our microscopes in. And they call that podocytosis. They've kind of settled on, these are a little bit easier to recognize, exo and endocytosis. Okay, while we're on the membrane kick, the cell can have outside structures that the membrane goes out and around. It's a projection of the cell. And there's a few different types of these. Now we're going to get into bits and pieces of the cell. We're going to start with the stuff that's projections of the cell itself. Pseudopods. Pseudopods are a distortion of the cell membrane that becomes like a big bleb off of it. So if you've got like an amoeba, is that amoeba might extend, the nucleus, might extend, it would flow into this stuff and push a piece out. Your white blood cells, a lot of your white blood cells move around tissues like this. Malignant cancer cells do this as well. And so originally people, they'd pick an amoeba up, they'd put it on a slide, and kind of, when you pick it up and move it around, it kind of gets upset and it kind of rounds up. And then if you watch it, you'd see this flow and you'd see this push out. And then the rest of the cell would kind of flow into that and be in a different spot. And it might push out this way and it would flow in that and be in a different spot. And they would look at this and go, well, it's like it's crawling. Like those are its feet. That it's stepping and then it's moving there and it's stepping and it's moving there, and somebody went, well, yeah, but they're not really feet. So what are we going to call them? Well, this looks really fancy. It is Latin for phony feet, for not real feet. Pod being feet, and pseudo being not really. People talk about pseudoscience, things that are represented as science but really aren't. So it shows up as in English words too. But that's a pseudopod. And actually, they can be very thick, they can be very thin, they can be used for crawling, they can be used for floating. Some things stick a whole bunch of them out, kind of helps suspend them in the water. It can be used for grabbing things. There's a lot of different functions that pseudopods have. Flagella and cilia are long, thin projections from the cell. And on a submicroscopic level, the level that you can see with an electron microscope, what they call ultrastructure, they're homologous. They're built with the same core. We'll talk a little bit about microtubules and stuff later. And there is the same arrangement of microtubules in flagella and cilia. So they're built the same way, but they're different in a bunch of ways. So I'm going to make two columns here. Flagella are bigger, cilia are smaller. And you go, so what's the cutoff? You know, where above the size or below the size? It's biology. The cutoff is kind of a gray area. You will find some fairly large things called cilia and some fairly small things called flagella. But in general, flagella are bigger and cilia are smaller. This other stuff is a little bit more trustworthy. It's not unusual to find a cell with one flagellum or two flagella or 4 or 6 or 8, but it's very unusual to find a cell with more than a dozen. Very, very unusual. Whereas you always find lots and lots and lots and lots, many, 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 many cilia. Sometimes the whole cell is covered with them, or they have bands of them with lots and lots and lots of cilia in the bands. So the numbers are quite different. His flagella are fairly rare. They're big. They don't need to have a lot of them. Cilia being smaller, you need a bunch of them to do anything with. When you got a big structure, you can modify it in different ways. There are flagella with combs. There are flagellas attached to the membrane like a big fin. There are flagella that are pushers and pullers that spin in only along part of them or do other. There's a lot of added structures you can find, not all the time, but if you're going to find add-on structures, you're going to find them on flagella. You're not going to find them on cilia. The closest you get to an add-on structure on cilia is that sometimes a bunch of them will fuse together and act like a single strut. But that's not really an add-on to a single cilium. That's combining a bunch of them. And when you got a bunch of little guys, you can do that. But flagella are big enough that there's a number of different add-on structures you can find on flagella. Coordination. If you got lots and lots and lots and lots of cilia, there's a potential for them to get in each other's way. So they tend to be organized in rows, and if the cilia are pushing this way in order to move the cell that way, or pushing this way in order to move mucus on your breathing tubes, then you don't want them hitting each other, you want them working together. And if you want to produce kind of a wave of motion, you don't want them all doing exactly the same thing. You want them slightly out of sequence with one another to produce this wave of push, And so they need to be very, very coordinated with each other. Whereas flagella, you only have a few of them anyway, and they don't necessarily need to have a lot of coordination in order to work together. Is that sometimes when you see multiflagellated cells, it kind of almost looks like they're getting tangled with each other. They're kind of just pushing the cell along, and there's not a huge amount of coordination on some of these. And then this one, in general, flagella typically work like little propellers. So they spin and they push water that way. And if there's a cell in the front of it, like on a lot of sperm cells, it pushes the sperm along. Cilia usually do this. They push and retract and push and retract, and there's thousands of them doing this, so they produce a motion that way. It's not always. You find flagella that do all sorts of other kinds of motions. You find cilia that do some sorts of other motions, but that's why it's usually. In general, you usually have a spinning stroke and a stroking, spinning motion and a stroking motion. Okay, one more cell projection that I kind of squeezed this in here so I could fit it on the bottom. Microvilli, little hairs, literally that's what this means, tiny hairs. These look like cilia. There's lots of little projections, but they're not homologous. What they have inside them is a different type of structure, and they can move, but they basically just wiggle. And what this allows you to do is take a cell surface that would be just that. Say it's a cell surface facing a space of some sort, and have all of these tiny little projections. Imagine this in 3 dimensions. Now that just took me a lot more line to draw that. That's because it produces a lot more surface. All of these projections out of your digestive lining, your cells along your digestive lining covered with microvilli. So instead of just having this surface for stuff to get through, it comes through all these other surfaces. And wiggling gives the, if these were really tight together and they didn't move much, then getting down in between them wouldn't be that easy for particles. But if they wiggle, it produces a little bit of an ability for particles to get even in at the bottom of the microvilli. And so you find these on surfaces where you need to move stuff through the surface. In our intestines, we're moving stuff in. Along linings of tubes in our kidneys, we may be moving stuff out. But it increases the surface area of the cell, and that's going to be a surface that you're moving stuff through. The direction depends on what you're trying to do, but it could be absorbing things, it could be secreting things, it could be moving stuff in or moving stuff out. Okay, that's basically the membrane stuff. Now we're going to go into the cell and look at internal structures in the cells. That's going to be the next section of this fairly long chapter.